I think I'm not going to say anything else, and I'll let you introduce yourself.
Okay, thank you so much. So yeah, as Mitch said, I'm interested in my PhD. I'm trying to understand brain size evolution in birds. And when we think about brain size evolution, evolutionary biologists find kind of a paradox, because brain size is a very expensive organ to grow in energetic terms. It takes time to grow, and it needs a lot of energy to maintain. So then, why investing in a big brain? Why some animal lineages, despite the big effort of having a big brain, they have these relatively huge brains? And there are many hypotheses out there, but in this study we focused on the idea that brains can be useful in changing environments. This hypothesis, the cognitive buffer hypothesis, was proposed some years ago, says that the brain could help animals to deal with changing conditions. If having a big brain allows you to develop new behaviors or adapt your behavior more rapidly to changing conditions, this should be selected in places where environments tend to change more often. We have indirect evidence that brain size is related to problem solving and innovation. So these kind of behaviors that could help you to deal with new situation. We also know that brain size helps to establish in novel environments, in the sense that introduced species that have bigger brains are better invasors. But there is no direct test on the relation between environmental variation and brain size in natural populations. So then there is another hypothesis that tries to explain why there is no evidence for the relation between brain size and environmental variation. It is the expensive brain hypothesis. This predicts that brains in places where there is a lot of fluctuation in the environment, because the brains need a constant intake of energy, a big brain cannot be afforded. So if there is long periods of food scarcity, animals living in these conditions cannot have a big brain. This is what expensive brain hypothesis predicts. So we have these two hypotheses relating environmental variation with brain size. Cognitive buffer predicts a positive relation, and expensive brain a negative relation. So in this case, we wanted to test this in birds to try to disentangle between the two hypotheses. And we thought that birds could be useful for that. First, because there is a big sample size on brain size data, but also because the birds live all over the world, so there are also striking differences in environmental variation. So we measured brain size in museum specimens. So there are big museum collections in some big museums in Washington, in London, and we can take the skull and fill it with some lead shot to know the volume of the brain, of the skull. And it has been shown that it's a very, very reliable measure to know brain size. So we had the brain of more than 1,000 species of birds, and also we only used individuals for which the body size of the individual was known. There are some specimens that we don't know the body size. So we only took the ones that the body size was annotated when the bird was captured or whatever. And we didn't use the damaged individuals. So then we can use body size to get the relative brain size, which is the measure we are interested in. So we did a regression between body size and brain size, and we took the residuals of this relation as the relative brain size of the species. Then we measured environmental variation using satellite data from first we took the distribution areas of each of our species from BirdLife and NatureServe. This is available, and we overlap these distribution maps with satellite data on enhanced vegetation index, which is a similar index as the NDVI of plant greenness. And also we took the snow cover. We had measures for every 15 days, something like that, for several years. But then we averaged for each species, we had a single value. For example, for this species, the chickadee, we had one value for the coefficient of variation of EVI along the year, between years or among years, and the number of weeks of a snow cover in their distribution area. With the species that migrate, so species that have two different distribution areas in summer and winter, we took the 3 winter months from the summer, well, 3 months in the summer and 3 months in the winter, and with this data we calculate also the coefficient of variation along the year, among years. But we didn't have data for migrants on the weeks of snow cover. So then in resident birds, we found that within year variation, among year variation, and the number of weeks of snow, all three are related to relative brain size in resident birds. And there is no relation in migratory species. And after we will see why. However, all these three measures, so these are 3 different models, and all these 3 measures are more or less correlated, so we cannot put it in the same model to see which of the 3 factors is more important, because we would have problems of collinearity. So what we did is to put these 3 measures in a PCA, and we find that the first axis they are all load in the same direction. So there is that explains 71% of the variation, and there is then the PC2 with 16% of variation. Explain it on environmental variation, and the first axis is related to more variable and harsh environments. So it's more or less related with latitudinal gradient. But then the PC2, we have negative effect of snow and positive effect of between year variation, and it's related to between year variation in lower latitudes. So for instance, like El Nino events and this kind of changes from year to year, and in places with no snow. This is what PC2 describes. And then we also use these 2 independent axes to correlate with brain size, and we find the same, that the more variation, both PC1 and PC2 are positively related to relative brain size in resident birds. Then we try to include confound factors to see if our results are confounded by other possibilities. And in a model selection with all these factors, we find that indeed the 2 factors with most important in terms of IC weight were the 2 axes of environmental variation. Then there are other factors that can contribute to brain size, but are not so important in our model selection. Then Well, here I show the best model of this model selection, where we see that both the two axes of environmental variation are positively related to brain size, but also diet breadth, like species with broader diets have bigger brains also, and species with longer incubation periods have bigger brains. But still controlling for that, we have this effect of environmental variation. We also include body size in the model, so what we are testing is the relative brain size change. We also check inside different groups of species, different orders of birds. We took the ones that for which we have a big gradient of environmental variation. So there are species in both higher latitudes and lower latitudes, and we found that in the strigiforms, passeriforms, and pisiforms, there is a significant positive relation between environmental variation and relative brain size, and there is no significant relation for galliforms. This is not surprising because these species mainly spend the winter months decreasing their metabolic rate, and sometimes they occur in a single pine tree eating needles. So compared to the other three, that they have an active search of food, we think that this could explain why this species doesn't have a relation between relative brain size and environmental variation. As I told you before, the three factors within years, no covariant among years, are related to latitude gradients. So these are three categories of low latitudes, mid latitudes, and high latitudes, and we see that they both increase in a latitudinal gradient. So this and this is what PC1 tells us. So we also check whether we have this effect in a latitudinal category, and we found that residents from higher latitudes have bigger brains than the other groups. And what about migratory birds? So all these results I have told you is mainly for resident birds, the ones that spend the whole year in the same place. We found no relation between relative brain size and environmental variation in migratory species, but we find a negative effect of migratory distance. So migratory species that travel longer distance have smaller brains. Even if there is no effect of environmental variation, it seems that traveling longer distance could be a constraint for their brains. So we had this short distance and long distance migrants. Well, we tested in the continuous of migratory distance and also categorization. And here we see how these migratory species avoid this decrease in resource availability in winter. So when we compare resident birds from different latitudes, we found that the ones in higher latitudes are the ones that have a biggest decrease in resource availability in winter. The same happens with migratory species in the place they breed, but because they travel to the wintering areas, they achieve to avoid this drop in resources. But this, as I have told you, have consequences in terms of the brain. So traveling longer distance is a constraint for them and they have a smaller brain. So overall, we find that environmental variation and migratory behavior can both affect brain size. However, we were interested in knowing if this is indeed the result of selection, so if brain size is an adaptive trait to these situations, or instead this could be related by different rates of evolution. There is evidence that migratory and resident species might have different rates of evolution, of any morphological trait. So we wanted to check if this was the case. And to do this, we have to do past reconstructions of the traits and test some evolutionary models. So basically, we are comparing two different models. The Brownian motion model predicts that once two species, two daughter species separate, the more the time passes, they have to be more different. They start to differentiate. So even if traits evolve at random, the difference between the two groups, two lineages, should be totally dependent on the time since divergence. So we see here that it doesn't care which, imagine the colors mean resident, migrant, or different categories. And here we have the phenotype. It's only dependent on the time since the divergence. However, if there is some selective pressures, some adaptive force, we should find something like that. For instance, if all resident species tend to have a similar trait, once there is a change in the behavior from resident to migratory or from one latitude to another, we should find a change in the phenotype, but also this phenotype should keep more or less constant if there is selective force to a particular phenotype. So we want to compare these two models in our data. We use as categories these 5 here. So 3 categories from resident species from lower, mid, high latitudes, and 2 categories for migratory species, short distance and long distance. Because this kind of summarizes the factors we have found that are important for brain size. So we reconstruct these traits in our phylogeny, and we found that there are several independent transitions between the categories, and this is the summary of in 1,000 different reconstructions, different simulations. This is the average number of transitions. So we see basically that, for instance, if we look at the residents from high latitudes, they can evolve from residents moving north to the higher latitudes, or from short distance migrants that change their migratory behavior and they became residents in these latitudes. But never directly from lower latitudes or from long distance migrants. What's interesting here is to see that long distance migration evolved 46 independent times from short distance migration, but never directly for resident birds. And then we tested these two different models, the Ornstein-Uklemek model of adaptive evolution compared to the Brownian motion model. And we find that the most supported model was an OU model. There are different, we can also test different Ornstein-Uklemek models, and we found support for the one that have different adaptive optima for the different categories. So this was the result of the model. So you find that the brain size optima, which is not exactly the same as the brain size of the species. It's like the brain size towards the species tend to evolve once they are in this category, is estimated to be higher than other resident birds. But still, even that resident birds from lower latitudes doesn't have to deal with big environmental changes like migrants, that they avoid these changes. We find that migrants have lower brain size optima than residents from lower latitudes, and it tends to decrease to longer migratory distance. So yeah, so as I told you, the longer the distance of migration, the smaller the brain. So here are the conclusions of our study. First, we found that the environmental variation is positively related to brain size in resident species. Then we found that migratory species have small brains, suggesting some energetic or developmental constraint of their lifestyle. For example, if because they have to move when environmental change, maybe they don't have enough time to develop a mature brain, a larger brain, before they have to migrate. So maybe they are, in that sense, we say that they could be developmental constraint. But also, if migration, for migration they need a lot of energy, maybe they cannot afford also another energetic organ. And finally, we found that the big brains are an adaptive trait in higher latitudes, as suggested from the Austen-Uhlenbeck models, resulting from selective process. However, we cannot say if this was selected once these birds colonized it, or after these birds colonized higher latitudes. We don't know that. It could also be that there is an environmental filter, so only species that already possessed a big brain or they were already adapted for these conditions, they were able to colonize higher latitudes after glaciation. So we cannot, with our results at least, we cannot disentangle between these two possibilities. And that's one thing I would like to do in the future. So now I'm trying to work in more causal models to try to see if brain size evolved before or after colonizing these latitudes in resident birds. And well, this is the end of the study, and we are happy because just last week we got accepted this study in network communications, and this is one of the chapters of my PhD thesis. So all the work in these 2 years, now it's worth doing. And thank you very much for all of you for coming to listen to this. And yeah, thank you also to Nacho to having me here this week to work. And many people that have helped me discussing many of these issues from Sol Lab in Barcelona, also from Nacho Bertomeu's lab. And thank you again for coming.